Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes, we have discussed the full analysis pipeline for transcriptomic data. So, we started with read mapping and we have talked about the specific requirements for read mapping in case of transcriptomic data sets, because we will have to deal with exons and introns boundaries, right. So, we can have splice variants. So, we need to have splice variant aware alignment methods. We specifically talked about few tools for that. We then discussed the quantification part and followed by discussion on the biases in RNA sequencing data. So, there are different types of bias that we might encounter when we are processing these data sets, and we also talked about some of the methods that we can use to actually remove or eliminate or minimize these biases. Followed by that, we talked about the normalization method. So, if we are doing comparison between different samples obtained from different conditions or different sample types, for example, disease versus healthy, we want to do a normalization before we can compare expression levels of genes between them. And then finally, we perform the differential expression analysis, right. So, we talked about the software tools that we use to do this differential gene expression analysis, specifically DSEQ2 and AGR, right. And finally, we talked about the interpretation of the data, how we actually visualize the results that we get, and we then talked about the functional enrichment analysis, right. This is interpretation of the results that come out of differential gene expression analysis. So, what we are going to do now is we are going to implement all those, right. So, what we will go through a hands on where we will actually analyze these real data sets, right. So, we will take one data set and we will analyze this data set through all these steps. Okay. So, that is the agenda, which is what we will start with this class, and subsequently few more classes will devote to this hands on. So, today's agenda will start talking about a programming language called R, right. So, this is a platform where we can do a lot of statistical analysis. A lot of the packages are built on this R platform, and we have talked about when we are talking about the analysis part, right. In the theory, we mentioned, right, there are packages that are implemented in R, and we will see, right, there are so many tools that you can use for doing this transcriptomic analysis. So, what is the goal of this hands on, right, for this next few classes is to perform differential gene expression analysis, taking a real data set and to identify genes that show differences in expression between samples obtained from two different conditions. Okay. So, this is just a specific example. Of course, you can take any example you like. I will also show you where you can get such examples or download such examples, and you can try your codes or try your hands at those. So, what we will do is we will start with this platform R today. So, this is where we will do all the analysis. Okay. So, briefly we have analyzed the transcriptomic data. I have shown you how to actually do the mapping with HiSET2 with a example data set, small example data set, and we obtain the raw counts using HDSEQ count. So, hopefully you remember that, and the rest. So, from that point we can now take this forward in the R platform. Okay. So, HiSET2 runs on the terminal of Linux, but from there on, right, also HDSEQ count we have used in the terminal. From there on we can now use the R platform to do all the analysis that we would like to do. Okay. So, just to remind you of the all steps, so summarize the steps. So, the first step is we have to set up the system in R. So, you might be familiar with R or might not. So, I will show you, right, at least very briefly how to set up this system, where you will get all the tools, etc., how to install these packages that are available in R, and how we can use these packages, where you get the manuals, etc., all the details. Okay. So, this is the first part, right. We have to get the system running, right. Then we will take the count data that we have already generated, and you can get this kind of count data, raw count data from different sites. I will again show you where you can get those. Then the first part of this analysis is the preliminary data analysis, right. So, you remember, right, we have talked about two different methods. One was the distance based clustering and the second one was the principal component analysis. So, these preliminary data analysis methods, they allow us to understand what would be the best comparisons to make, okay, whether some samples are similar to each other or whether some samples are different, right, and it can identify the best groups for comparison. Okay. So, we will go through this preliminary data analysis with our data set and then we will also do the bias correction part, right. So, we have the count data. So, we will do the bias correction again using an R package that can allow us to do some sort of bias correction, and then we will actually go to the differential gene expression analysis. This is using DSEC2. Okay. So, we will try two different methods. We will do with DSEC2 first and then we will also try this differential gene expression analysis through a RNA-seq method, right. So, where we will utilize these ERCC spike-ins to actually normalize the data. Okay. So, if hopefully you remember what these spike-ins are, we have talked about this and how they are we can utilize those spike-ins to normalize the data. Okay. So, we will go through these two methods. Okay. And then we will have the results and visualization part. So, once we have generated the results from differential gene expression analysis, we will talk about how to visualize, right. So, we will see what kind of code we can use, right. So, I have shown you some of the visualizations, but we will here in this hands on, right, we will write these codes to generate those visualizations, right. For example, the MA plot or the heat map. So, those we will do here hands on, and then finally, I will also talk about the functional enrichment analysis. There are different tools that you can use, but we will use one tool to actually do the enrichment analysis from our data set. Okay. So, this is the agenda. So, let us start with the first part, which is the setting up the system. So, we will have a brief introduction to R, how to set up R and very basic commands in R, and then followed by the setting up the system for the data analysis. Okay. So, and then if time permits, we will also talk about the data loading, right, and we can load the count data and we can look at some of the characteristics of the count data. Okay. So, let us now move on to the system, right. So, I will start with the R platform, right. So, here it is. Okay. So, this is the website where you can actually get this tool, right. You can download R for your system. So, R is a free tool that actually is used for computing for and also generating very nice graphics, and it can run on all three platforms, right. So, it can run on Unix based platforms, Mac as well as Windows. Okay. So, if you are running Windows, you can simply choose the right version. So, if you click on download R, you have a lot of these options, right, different methods from where you can download, or you can simply go here, right, and the newest version, and you can download the newest version. Okay. So, here is the .tar.gz file we have talked about, right. These are the compressions and you can download them and decompress them. Okay. So, I have installed this R 4.3.0. Okay. So, as you can see, 4.3.1 is the latest one, but I have in the system is the 4.3.0. Okay. So, we will utilize this. You can use 4.3.1. There won't be too much difference in terms of use of packages and the some of and the updates, right. Okay. So, let us look at this. So, if you want to install, you can download this, right, and install for your system. Again, you can use different right, whether you need Windows system or so for Windows release, you can actually go here, right, and you can see, right, all these methods, etc., all these things. Okay. All right. So, I will show you probably. You can go here, right, and again you have this download R for Linux, right. So, if you are running this Linux systems, you can download from here. If you are running Mac OS, you can also download from here, and for Windows, you can simply click and it will download all the tools for you. Okay. So, some of the tools are required along with this base, right. So, you can see the base, right. This is done when you are installing R for the first time, and also you need to download this R tool especially, right, and some of the CRAN packages you probably want to download as well. Okay. So, if you do not download these packages for R, we can install them later on from R, from inside R, okay, and I will show you how we can do that. For Linux system, if you are running Linux based system, so I can show you in WSL, right, the Windows subsystem for Linux, and here you can also install by simply saying sudo, if you have sudo permission, of course, install rbase. Okay. So, in my system it is already installed. So, in this WSL system, so I will not run it. So, in the Linux system, if I just want to run R, I will just type capital R and then I will enter, and you can see here the version is different, right. You can see this version here, this is a slightly older version, but it is installed here. And if you can write your codes, you can generate all sorts of plots, whatever you want, by simply typing here. Okay. So, you can write your commands, for example, you can say equals to 3 or something, right. You can run all sorts of commands here, and you can generate all the graphs or do statistical analysis, whatever you want inside this terminal. If you want to quit, right, if you want to come out of R from here, you can simply say Q brackets, right. So, and then you can say whether you want to save the workspace image or not, usually no. So, we are back to the terminal again. Okay. So, it is that simple, but if you have a very long R code to run, let us say you have generated a very long code of R, then you do not probably want to run it this way, right. You have to copy and paste all those things. So, that is very cumbersome. So, you can run it as a script here. Okay. So, you can simply run, let us say R and then -f and then script name. Okay. So, this is also possible. We will not get there because we are not going to write very complicated scripts. I will show you small scripts one at a time, and we will run those and execute those, and we will generate some of the results. For this class and demonstration, I will use the Windows version for a very simple reason, because if I run this from this Windows subsystem for Linux, all the codes, the plots that will be generated, that will be saved in files. Okay. So, I have to always save them. I will not be able to visualize them here because it is not set up like that. So, that is why I will use the R from Windows, because then I can see the side by side the plots that are generated. Okay. You can also download something called R Studio. So, I also have this installed here. Okay. So, this is the R, I have it installed, this is 4.3.0, and I will run everything from inside here. Okay. I can probably zoom in a bit, let us see. I can make it a bigger, slightly bigger, I think, and then we can probably see this better, and here it is, right. Okay. So, this is bigger now. Okay. So, once I run the codes from here, the plots will also be generated side by side. Okay. So, I can see the plots in this part. So, that will be very useful, right. So, I can see and change if I want to change the plots and the outcomes that we see. There is also something called R Studio, right. This is also very helpful for running our codes, because in here you are more aware of your environment. Okay. So, what you have is on this side you have the global environment, right. So, it means which variables are there in the environment, right, what those values are, etc., will be given here. So, maybe if I just write, for example, okay, maybe later. So, if I let us say write a equals to 3, you see this is there. Okay. So, a equals to 3 is now in the environment. Okay. So, that is in there. I can remove this from the environment by just saying RMA. Okay, remove A. So, this is gone from the environment, right. So, this is also possible for other types of data structures in R as well, right. So, we will talk about that later. On the right hand corner here, you have these plots that you can see. You can also see the packages that are already available in the library and which are installed, right. Of course, you can see some of them are ticked, right. So, it means they are also available and installed, right. and this is very helpful because then we can see the plots around here, right. So, whatever plot I am generating, I will be able to see this here, and here we can write the code, etc. Okay, so this is the console. You can also see a terminal, right. If you want to use the terminal here, you can also use it from here. Okay. So, for keeping this simple now, we will use the just the R, okay, not the R Studio. Once you get more familiar, you can start using the R Studio. Okay, so I'll quit this now. All right. So, now going back to R here. Okay. So, R can support a lot of different types of data types, right. So, you can have lists, you can have vectors, you can have matrix. So, different types of data can be used in R, right. So, to create a vector, you can simply assign a vector, right. So, let us say we have a variable name that we say it is vector, okay. And to assign, we usually use this arrow sign, right. We are assigning these values to the vector, okay. But you can also use this equal sign, okay. It is simply we are assigning these value in vector, okay. So, you will see both of these, right. You will see this assignment, okay, as well as the other type, okay. So, vector is a list that contains objects of same type, right. So, it can be string, it could be number. So, I hope you are familiar with the number, string, etc., those concepts, right. So, you can have, let us say a, right, or character simply, right. And to see what is there in vector, you simply type vector, okay. So, this is what we see. We can also define list, right. So, let us say we say list lst and list can contain different types of objects, right. So, it can contain numbers as well as the strings or characters, right. So, it is kind of like a mixed type of thing, right, and you just want to see, right, what is there, you say lst, okay. Now, if you want to access these elements, right, so you can see these numbers, right. So, lst is 1, right, and 2, this is the third element is d, right. But if you want to access these individual elements, right, you can simply write and it will give you these elements, okay. So, the other one you can write is this and you get this element, okay. You can also define matrix in R, right. So, this is the type of data that we will encounter most. You will see we will work with mostly matrix data because we are talking about count data, right, in transcriptomic data. So, round data means we have genes in the rows and we have the samples in the columns, right. So, this will be a matrix, okay, which will again the dimensions will be determined on the number of genes that you have in the sample and the number of samples, right. So, we will come to that in a moment when you actually start working with the read data, okay. So, you can say matrix or as matrix, right. So, you can have, let us say so, we have to mention, right, how we want to distribute this expected comma, I think is the data, I think, okay. So, we need to give C, right. So, it will say, okay, this is the value, these are the values that we have in the matrix and then number of rows is 2. So, we have 6 elements, right. So, you just have to tell how you want to organize these elements in the matrix, right. So, here I want, let us say n row equals to 2 and n column equals to 3. So, 3 columns and 2 rows, okay. So, let us now see, okay, this is how it is organized now. You see like we have 3 columns and 2 rows, okay. Now, how to access these elements in this matrix? We say 1 comma 2, right. So, the first number is for the row, right, and the second number is for column, okay. So, this will be C is this element, right. So, 1 2 will be this element and similarly you can try other ones, right. So, you can say 2 3 This would be the last element here. We can also try something like this. Okay, this when I just say 1 comma gap, so it will give it will mean that we want the full first row. Okay, so that's what we are getting here, right? We are getting the full first row, and similarly we have, if you let's say want the last column, full last column, so we can say gap comma 3, so we will get the third column. Okay, the values in the third column, which are E and F. Okay, so remember this matrix part, because this is very important. This will actually come when we start loading our data. There is another concept which is called the data frame, which could be combinations of all these different data types. Okay. All right, so I think this is clear, right, how we actually get to the different types of data, how do you actually start working on this. Okay, so what I will do now is I will load the data that we have, right, the raw count data that we have generated, and we will start performing some operations on this data set. Okay, so we will start loading the data first, but let us have a look at the data. Okay, so for that I will go to the terminal, because here I have loaded the data. It is here inside this folder. So this is the ingest data analysis hands on test, and this is the file that actually contains the raw count data. Okay, so we can check this, right, in the terminal using vi. Okay, and one thing I will do first, hopefully remember the commands in vi :sc node app. So what you see here on top are the sample names, right. So you have these S1, S5, S9, so on. So we have 16 samples up to S16. Okay, and along the rows we have the gene names. Okay, so you can see the gene names. We can check how many columns or how many rows we have. So if I just go to the end of the file, right, I get 6,806. Okay, so in total we have 6,806 values for genes, etc. Right, and then for each sample you have the count value, right. So you can see this for this gene YMR056E, right, we have these count values across these samples. Okay. Now these samples are organized in certain ways, which will come about later on when you actually go to the differential expression analysis. That is when the whole thing will be more relevant, right. So how these samples are organized, which are the replicates, etc., that information we will talk about later on. Okay, so as you can see, this is a matrix, right. So we will like to load this in R, and then we will start working with that, right. So to load this in the R, right, so we will again go here, right. One of the things we have to do is we have to give the path to the file, right. So we'll only then we can load the file, right. So to get the current working directory where we are, right, inside R, we can use the command called getwd. Okay, so getwd is the current working directory, right. So working directory. So it gives me the path where we are right now. Okay. Now to change this to the directory, right, where we want to go, right, we have to say set working directory or setwd. Okay. Now in your system these paths will be different, of course, right. So you can then go around and go to the appropriate path where you are getting these files. Okay. So you say setwd, I know where it is, right. So it's on, if we go up, right, so it's again you can see that this is like Linux commands we are using, right. So // means we are going back up, right, one directory up, then we are going in desktop where I have the data, right, and in hands on I showed you, right, where this data is, and that is it, right. So this will change the directory. It's like cd, right. In terminal we use this cd, here we just use the setwd and within double quotes, right. The whole path should be within double quotes. Okay, this is the format in R. Okay, and we can again check whether we have changed the directory and we are in the right directory. We are, right. So the next thing we want to do is we want to load the data. Okay, so to load that, we can load a matrix as a table, and the command that we have in R is called read.table. Okay, so the first thing is we need to define a variable where do we want to load the data, right. So, let us say we say call it as tab, right, on the table for the short for table, right, and then read dot table, and we have to now give the file name, okay. So, which is this one, okay, and again this file name should be within double quotes. The other parameter that we need to specify often is whether we have a header line in the file or not, okay. So, header equals to true, right, this is a logical variable, right. So, header equals to true means there is a header line on top of the file, okay, and we need to consider that, okay. So, you can try this without header and with header and see what the outcome is, because this will impact, right, how we actually call the data, okay. So, we have a header line, right. So, I showed you we have these S1, S5, S9 etc., those names, right, in the first line. So, that is the header line. So, that is why I will mention header equals to true, okay. So, this is what we get now, header equals to true, and we can now actually simply type tab. So, it will print the whole data here, okay. This is something we do not want. What we want to do is we want to check, let us say, first few lines, okay. So, we can simply say head tab. So, head will again show you first few lines. So, you can see this is very similar to the command lines, right. So, the terminal what we have done before, right. So, you can simply say head tab. So, it will show you this part of this matrix, right. So, you can see these first 6 genes here along with the header that you have, okay. So, the next thing, right, so you can also try to see the last few lines. Again we can use the tail, again is very similar to the command line. You can also specify number of lines you want to see. You can say I want to see first 3 lines, okay. So, you say head tab 3, so it will show you only first 3 genes. If you say head tab 10, right, you can see first 10 lines, okay. So, this is how the whole thing would work. Now, let us say you want to see, right, now the difference will come is, let us say we want to see the first row, right. So, we can see YMR 056C because this is stored as a matrix, this data, right. You can now access each element, you can access rows as well as columns, okay. So, now the next thing is we can access the columns also, right. So, let us say we want to say tab 5, right. So, this is the 5th column. Of course, we get all these 6,805 data points, right. So, maybe you can say we want the 5th column only the head, right. So, first 5 or 6 values we get here, okay. Now, it turns out in R you can actually access the columns in a different way as well, and that is actually very convenient, as we will see later on. So, I will just type head tab again, right, and look at the column names, okay, S1, S5, S9. So, in R when you are dealing with really big data, you can actually access the columns by this tab dollar the column names, okay. So, I will say tab dollar S5, okay, and it will give you the values corresponding to S5 column, okay. So, again I have used head, so, we will get the first 6 values here and these are the values, okay. You can see this is what you have under this S5. So, what is really convenient here, you do not have to remember, right, which column contains which sample. You can simply call by the sample name or the variable name, right, and see what the data is, or you can do all sorts of analysis on that, okay. So, this is actually really convenient for while you are using R. Okay, so, then you can also generate something called summary tab etc., okay, and it will give you the statistics for all sorts of for all the columns that you have in the data, okay. So, you can get this minimum value, you have the first quartile, you have the median, you have the mean, you have the third quartile, the maximum value etc. So, for each sample S1 from S1 to S16, you will get these statistics when you write this summary. You can also see, right, what are these variables that are available using this dollar approach, okay. So, you can simply type tab dollar and it will say S, and it will then show you what are these variables or the names that are available with this dollar access, okay. The other option that you have is you can also type names, okay, tab, and it will show you these names or the column names that are actually available. Okay, and you can also try row names, I think, and you get these names of the genes that are available. Okay, so you can try these column names or simply names, and you can also try row names, right, and this will give you the list of genes that are present in your data. Okay, so I think this part is clear. So if you do not have your own data, right, I'll show you in the next class where we can actually download this data, right, and we can try this out on these different things on them. Okay, so if you have your own data, definitely try this and try to load it, and then you can start working on this. So in the next class, we'll actually do a bit more on this data set. Since we have loaded it now, we'll actually now we can do a lot of things, and I'll also show you how to start loading the packages that will be required for our analysis. So that's it for this class, and thank you.